This question is about how chloroplasts are specialised for photosynthesis. So we've got to describe in detail how chloroplasts are specialised. So just talking about the structure of chloroplasts, first of all, they've got a large surface area. Similar to red blood cells, they are biconcave in shape, and it's that biconcave shape that provides a large surface area for the absorption of photons of light. Also, in terms of the structure, they've got grana or stacked thylakoids, and they're stacked like that. Again, that provides a large surface area. Now, these thylakoids are the site of photosynthesis. More precisely, the light-dependent stage. These structures contain chlorophyll, which acts as a transducer. So it converts light energy, and that chlorophyll absorb light. Don't say catch light. Don't go out and catch it. Absorb light. Right, and chlorophyll absorbs different wavelengths of light, mainly red and blue, which is why chlorophylls are green, because they reflect the yellow and green parts of the wavelengths. So they absorb different wavelengths of light. They're arranged in photosystems. So at the center of the photosystem, you've got chlorophyll A, which is the primary pigment, but you've also got accessory pigments in the photosystem. We've got xanthophyll, carotene, and chlorophyll B. These pigments funnel electrons to the reaction center, and this part, this is the reaction center. So photons of light are absorbed, electrons are excited, the different chemicals, the accessory pigments, funnel the electrons to the reaction center, where you've got chlorophyll A. You've got two different photosystems. You've got photosystem 1 and photosystem 2. And they have different absorption peaks. So they absorb wavelengths of light that are slightly different. So once you've got your membrane, you've got your photosystems. So you've got photosystems, and between the photosystems, you've got an electron transport chain. And these are made of electron carriers. And they carry electrons down that electron transport chain. The energy, as the electrons drop down in energy, and don't forget there's 2 electrons that are promoted from chlorophyll, you've got chemiosmosis. It's used to pump in hydrogen ions from the stroma, from the stroma to the thylakoid space. So you end up with a high concentration of potassium ions, hydrogen ions inside of the thylakoid space. So that acts as a proton pump. You've then got an enzyme called ATP synthase. And these hydrogen ions will move through ATP synthase. And you've got the production of ATP. So you've got phosphorylation taking place, and phosphorus is added to ADP to make ATP. At the end of this chain, the electrons are picked up, and you get NADP H produced from NADP. So you've got NADP present in the stroma. You can write NADPH2. Right, those products then, so the ATP that's produced and the NADPH that produce, then enter the light independent stage. And again, we're concentrating on the light specialized. So in the stroma, there's enzymes. That are linked to the light independent stage, and this enzyme is called rubisco. Don't forget the light independent stage is called the Calvin cycle. Again, just talking about the structure, how it's specialized. So within the chloroplast, you've got the storage of starch. You've also got ribosomes and DNA for making proteins. For example, the enzyme rubisco. And that covers everything to do with describing in detail how chloroplasts are specialized for photosynthesis. This is a copy of the mark scheme, just so you've got a copy. You need to make sure on top of this that you understand exactly what's going on the light dependent stage and the light independent stage.